Like people are still filtering in. We've got about 54 attendees right now. But we're going to go ahead and get started so that we can have these amazing panelists share their presentations and tell you a little bit more about this. My name is Jeff Lebo. I work with NF Network. I am their development director, and I'm here in place of Kim Bischoff, who is the executive director of NF Network. We're very honored to welcome our panelists today, Dr. Paul Moots, Dr. Colleen Niswender, and Mike Sager from Alexion, as well as Harrison Parent, who works with Dr. Niswender. So we are here today to talk a little bit about the value of and the incredible work that's happening in NF research and clinical trials. One thing to note is that a couple of years ago, NF Network actually began funding its own NF research, and we're very excited about the prospects. We've already been doing this for a few years. Thanks to a project that I work on, we were actually able to completely fund this next year of research as well. So if you would like to support NF research through NF Network, it's a project called Project Dream, and I'll make sure that we put a link in the chat that you can have access to, or you can visit our website, nfnetwork.org. So it's an honor to have our panelists today, certainly not about us. So first I'd like to introduce Dr. Colleen Niswender. She's from Vanderbilt University and the Warren Center for Neuroscience Drug Discovery. She and her lab have begun working on NF research, and they're outlining drug discovery process. They're working anywhere from compound identification and screening to clinical development. And we're honored to have her here to tell how these efforts are further advancing the treatment pipeline. Dr. Niswender, welcome.
Sure. Thanks very much for having me. I'm here with Harrison, who is a fourth-year graduate student in my lab, and we're going to kind of share this presentation. And I will begin, and I'll tell you a little bit about some of the things that we're doing at Vanderbilt that are kind of unique for an academic university. And then we'll tell you about how we got into neurofibromatosis type 1 research. So I have been at Vanderbilt. I went to graduate school at Vanderbilt, and I got my PhD in pharmacology. And I came back to Vanderbilt after my postdoctoral fellowship at University of Washington in 2004. And I joined what is now called the Warren Center for Neuroscience Drug Discovery. So the Warren Center was kind of born out of the idea that neurological and psychiatric diseases are very difficult to attract pharmaceutical industries to invest in because they tend to be really hard. And so the goal of the center was to try to do a lot of the de-risking that happens to try to push an idea from an idea that a scientist would have in the lab. How do you get that to a place where you can then do a clinical trial and test that in a person? And so what you're seeing here, we were endowed in 2020 by the Warren family. And so that's why we are now the Warren Center for Neuroscience Drug Discovery. And so you see profiles of the leadership of the Warren Center. And so the mission is to then translate advances in basic science. And these are often coming from basic science labs at Vanderbilt University and Vanderbilt University Medical Center. To novel therapeutics by de-risking innovative approaches for treatment of brain disorders. And so we've kind of assembled in the center, we have it fluctuates 50 to 60 full-time employees within the Warren Center. And actually, if you've ever been to Nashville and been to the Vanderbilt campus, the main Warren Center is actually about 20 miles south of Vanderbilt. I tell people it's out by, there's a Burger King next door, and there's an Academy Sports. But it's a very nice like biotech. And if you go there, it looks very much like what you would see in a biotechnology company. And so we have all of the major infrastructure that you would see. Actually, we have more than often biotech companies have because we can leverage a lot of our assets with resources at Vanderbilt University. And so we have been very successful over the past, I've been here since 2004. Like you might see in other drug discovery or biotech companies, we actually have a pipeline of products that we are working on. And these include, we just started a phase 2 clinical trial for Parkinson's disease. And actually for that program, we started a company here in Nashville that's called Acadia Pharmaceuticals. And then we have a number of other projects here. All the writing on this slide isn't really necessary, but it's just to give you an idea of what we do is we develop compounds to a stage, and these are small molecules, develop compounds to a stage where we can partner with a pharmaceutical company so that we can do a clinical trial in patients. And for me as a scientist, you know, I mean, there are a lot of scientists who work their entire lives on something that they're super passionate about. They work on it in the lab and it's very specific and they love this target. And we love our targets too, but I think that those of us in the Warren Center, we have this drive or passion to be able to translate that into people. And so can we do things that we move new therapeutics into the clinic? And so we've been successful in that we now have 6 compounds, 6 drugs that have come from the Warren Center that are now in clinical testing. And as you can see, some of our areas are, obviously we're neuroscience focused. And so we've been very focused on things like schizophrenia, Alzheimer's disease, LBD stands for Lewy body dementia, cognition, Prader-Willi syndrome. This is a new project in the center, sleep apnea, migraine, et cetera. So we're on target for 4 to 5 new phase 1 clinical trials this year. So we're very excited about all these projects. So how did we get into neurofibromatosis type 1? And this came from an effort that Vanderbilt University Medical Center has assembled a database that is called BioView or BioView. And basically what happens is this is a collection of de-identified patient DNA samples. So if you are a patient and you come to Vanderbilt, what they will do is if you have to go to the lab, let's say, is they will collect blood sample, for example, and they will bank this here at Vanderbilt for investigators to then perform clinical level research. And really research in people is really what trumps everything. And so if you can have clinical data to support new drug development, for example, or some type of new therapeutic, then that really grounds your research in a place of translational relevance. So your participation when you come to Vanderbilt is fully optional. You can opt out of this. So for example, I've been to Vanderbilt many times for many different things. I'm in BioView. However, I don't know where I am in BioView because everything is tied to de-identified patient records. So I can't find myself in BioView. And actually what's really interesting, one of the things that they do is they actually shift all of the dates in BioView. So let's say you saw the doctor on October 3rd for diabetes or something. Well, in the de-identified record, your date that you saw your doctor would really be April 15th, something like that. So you can't trace things back to an individual patient. And so there's about 300,000 samples that are banked in BioView. And it's one of the largest resources of its kind in the world. And it actually served as the basis for something that's called All of Us, that is happening at the National Institutes of Health, which is a global repository of many more samples. The other thing that's in BioView is your whole medical record is in there. And so what we can do is we can take what's called phenotypes, so a symptom that's in BioView, and we can say, are there genetic correlations with any type of gene? And so for us, we're very interested in particular types of receptors in the brain that we are trying to develop drugs for. And so the BioView people came to me and they said, we really want to work with the Warren Center and we want to be able to use data in BioView to give you guys new ideas of things to work on. And I said, okay. So I have been working on a receptor in the brain that responds to glutamate, which is an excitatory neurotransmitter that is called metabotropic glutamate receptor 7. And I had been very interested in this receptor for a very long time. And what you see is that people who have, it's a neurotransmitter receptor. It's important for normal cognitive development and function. And what has happened over the past 10 years or so is that there's been reports in the literature of people who have a primary mutation in this gene, in the GRM7 gene. And they have very, very severe neurodevelopmental phenotypes. So they have seizures, they have intellectual impairments, they have hypotonia. defects in myelination in their brain, et cetera. So we had been working on this particular receptor from the status of other neurodevelopmental disorders, one of which is called Rett syndrome, and we do a lot of work on Rett syndrome. So the BioView folks came to us and they said, hey, let's see what other symptoms are associated with this gene in BioView. I said, okay. So their analysis that they do, which they call Phenome-Wide Association Study, or PheWAS, identified an association between our gene and neurofibromatosis type 1 in BioView. So clinical data that said somehow GRM7 is somehow linked to NF1. Now we know NF1 is caused by mutations in the neurofibromin gene. So we don't think mutations in GRM7 itself cause NF1, but we think that there are correlations between what are called single nucleotide polymorphisms. These are just changes in your DNA. Everybody has thousands of them, but that there are SNPs in your gene that somehow are correlating with NF1. And so we use these SNPs to make correlations between genes and various diseases. And I'm going to turn it over right now to Harrison. But before I do, I'll tell you a story about this project. So we started working on this project and we were very interested in, so our gene we know is involved in learning disabilities, ADHD, cognition, seizures, etc. And so when the BioView people came to me and they said, we have a correlation with NF1, I had traditionally thought of this as a tumor disorder, and I don't do cancer research. And so I wasn't quite sure how to address this correlation. And so I went over and I talked to Dr. Moots and I said, hey, I have this gene. It's involved in cognition and ADHD. And he said, well, that's interesting because there's a large percentage of patients who have NF1 that have learning disabilities and ADHD, but we don't know why. It doesn't seem to correlate with how many tumors they have. It doesn't seem to correlate with what their mutation in neurofibromin is. But we think, so we started talking about the idea that maybe there was a modifier gene that if you had certain SNPs in our gene, GRM7, maybe you also have NF1, then maybe you are more likely to have learning disabilities and ADHD. And so we started studying this and I recruited Harrison into the lab. And so he took over this project and we were doing things that he'll tell you about, that were very sort of more in the basic science realm. And he gave a talk at an internal conference at Vanderbilt. And a student came up to his poster and she said, hi, I'm Emily. And he said, hi, I'm Harrison. And she says, I happen to work on learning disabilities in children with neurofibromatosis type 1. And we were like, what? So it's kind of an example of the serendipity of science and being in the right place at the right time. And so this has led to a number of collaborations with Lori Cutting's group here at Vanderbilt University Medical Center. And so I'm going to turn it over to Harrison and let him tell you about his project and some of the things that we're finding. And I'm happy to answer any questions at the end of the presentations.
Great. Thanks, Colleen, for the introduction. My name's Harrison Parent. I'm a fourth-year graduate student in Colleen's lab, and I'm actually in the PhD program in pharmacology. So I'm in the same program that Colleen was in when she got her PhD at Vanderbilt, and now I'm working in her lab. So it's really interesting little full circle moment. But I'll talk to you about my project in the lab, which is looking at NF1 and this correlation between NF1 and mGlu7 that Colleen was just talking about. And so this slide here, what we have up now is some data from a clinical population. So we have about 530 kids that have participated in some clinical trials that Dr. Lori Cutting's group was running at Vanderbilt. And what we saw here was we take these SNPs, or these single nucleotide polymorphisms, that originally we saw were correlated with NF1. So they have these two names, RS779710 and 9870680. Those numbers aren't really important, but it just sort of delineates the two that we found this correlation with. And what's interesting is in this population of kids that actually does not have NF1, we've seen that if you have what's called the minor allele, so you have this base pair in your DNA, and if it's mutated and you have this SNP, if lower than 50% of the population has, say, a C, and greater than 50% of the population has a T, the C is considered what's the minor allele versus the T is the major allele. So what we've seen with these two different SNP sites is if you have two copies of the minor allele, that's actually correlated with reduced full-scale IQ scores, as well as reduced scores on these two different readings on the right you see, which measure something called phonetic decoding, which is essentially they give a sort of an assessment to these kids, and it has these words, but they're not real words. They, follow normal spelling rules and things like that in English, but they're not actual words, and they can test how do the kids sound these out. And it's kind of a measure of, word learning and reading learning. And we can see that if you have two copies of the minor allele, you also have reduced scoring in these cognitive tasks as well. So this is really interesting because looking at NF1, Colleen, if you can progress the slide for me, looking at NF1 for the sort of the cognitive phenotypes that we see.
Sorry, it's not progressing. I don't know why. It was doing fine. Oh, here we go.
Okay, great. So yeah, looking in NF1 at the cognitive phenotypes that we see. So it's been reported that up to 80% of NF1 patients can have some sort of cognitive phenotype. And like Colleen was discussing, this can be ADHD, reading disabilities, and things of that nature. We've seen like what I was just talking about with full-scale IQ. You can see on the right that there's been a previous study with a large group of NF1 patients that saw significant reductions in full-scale IQ scores compared to kids that were tested that did not have NF1. And there's several other domains that they tested as well. So it's really interesting that we see these decreased scores in kids with these GRM7 SNPs as well as kids with NF1. So it could be a potential overlap here with the cognitive phenotypes of NF1 and GRM7 SNPs. And so, like was previously mentioned as well, the cognitive symptoms don't seem to align with the other disease-related symptoms. And that also means that the approved therapies, so the selumetinib and mirdametinib that have been recently approved for plexiform neurofibromas, they don't really work for the cognitive symptoms of the disease. So they really lack any approved therapies that are effective for them. And so here I have a little diagram of a neuron. So I'm going to use this to kind of describe what this project is sort of getting at. So here, we have the neuron, and you can see mGlu7 up at the top. So that's the receptor protein that the lab has been working on. And down at the bottom, you have neurofibromin, which is the protein when it's mutated and you have loss of its function, it gives NF1. In the middle there, you have synapsin-1, which is a protein within neurons that binds to those little bubble-looking things there on the left called synaptic vesicles. And those vesicles hold neurotransmitters, which are what your neurons use to communicate with one another. Now, when those vesicles are bound to synapsin, it retains those vesicles, keeps them away, and prevents them from being released. And so what happens in NF1 is if you lose function of neurofibromin, this protein sort of can act as an inhibitor of this molecular switch called phosphorylation. And when this protein synapsin-1 is phosphorylated, it releases those vesicles, and then those vesicles can then release their neurotransmitter when the neuron is stimulated. So what happens in NF1, you lose function in neurofibromin, you lose that braking mechanism. You get increased phosphorylation of this protein synapsin-1, which leads to increased neurotransmitter release and contributes to those cognitive impairments or learning disabilities that we see in NF1 patients. Now, our idea here with this project is that we have compounds that can target this protein mGlu7. They can either directly activate the receptor or they can sort of enhance its activity. And mGlu7 can also act as a braking mechanism for this synapsin-1 phosphorylation. So our idea is if we can sort of activate this receptor or increase its activity, we can compensate for that lost braking mechanism with neurofibromin mutations, reducing neurotransmitter release and sort of restoring what would be the sort of normal level of neurotransmitter signaling.
Sorry, Harrison, the slides are not advancing. I don't know why.
No problem. That's fine. Oh, here we go.
Oh, here we go. Sorry. I don't know what's going on.
No worries. So building off of that, like I mentioned, we are hypothesizing that by increasing synapsin-1 phosphorylation, in NF1, we're driving these cognitive symptoms. And if we can target mGlu7, we can reduce that phosphorylation. That will potentially correct those cognitive symptoms. And we've actually seen in the lab, so this is looking at intracellular signaling of these neurons. So looking at that synapsin 1 protein, looking at that phosphorylation directly. So you can see on the right in that graph, if we add an mGlu7 activating compound to these neurons, we can significantly reduce the amount of synapsin phosphorylation that's present. Now, this is a really, interesting step forward. We're kind of trying to build off this now too and see if we can directly measure release of those neurotransmitters that I was talking about. It's something that we're working on right now in the lab. But we think that, with this step, by correcting this synapsin phosphorylation, we can sort of try to change the larger scale function of the neurons to correct the phenotypes or the symptoms that we see. And moving sort of from those individual cell signaling pathways into more like neuronal circuit-based signaling here. So we have animal models, mice of NF1 that we use to sort of study, this neuronal signaling as well as some sort of cognitive tasks that we give them. And on this slide, I'll talk more about that circuitry. So what you can see on the right here is a graph where we have slices of these brains that we can stimulate with electricity. So neurons respond to electricity, and that's kind of how they signal with one another along with those neurotransmitters. And if you give a small electrical stimulus, you can see on the left side of that graph, that line near the bottom at 100 is sort of their baseline signaling. So if you stimulate with a small amount of electricity, you get a certain response from them. At that 0-minute mark, what we do is we give a really strong, really rapid stimulus. And afterwards, we then go back and give those small stimuli again. And what you can see is that those neurons then, to the same stimulus as before, they respond much stronger. And we call this plasticity. So plasticity is a really important characteristic of your neurons, and it's essential in learning and memory and things of that capacity, and particularly within the hippocampus, which is a region of your brain that's really dedicated to learning and storing those memories. So you can see on the right, now this was not done by our lab, this is done by a different group several years ago, but what we can see is that NF1 heterozygous mice, so this NF1 plus minus, that black line is lower than the white, which is wild type is the non-NF1 mice. So that having NF1 has a significant impact on this plasticity that we see. What's interesting is that we've done studies very similar in a way, is that we have slices, brain slices, and we have that same sort of baseline. So you can see that line there to the left of 0 on that bottom graph. So we're stimulating those neurons. They're giving us a response. We give a smaller, it's still a fairly large stimulus, but it's slightly smaller than what we saw above. And we get some sort of plasticity, but as you can see in the black line, it comes back down and reaches that baseline again. Now, what we've done is we also treat these slices with this compound called VU288, which is some compound or a drug that can bind to mGlu7 and sort of increases its activity. And what you actually see is if you treat those slices with that compound VU288, you can increase that plasticity response at a long term at about 60 minutes. And that's really interesting because we have that small reduction as we see above in the NF1 mice. So our idea here is that if we give this drug to those slices from NF1 mice, we can correct that plasticity level to what is in those non-NF1 mice. And the idea with that is if this works, that might be sort of what's driving those cognitive impairments, and that we might be able to correct them with that. So yeah, like I mentioned, we're currently testing that. And then, oh, sorry, you can go for it.
Mm-hmm.
And then lastly, I'll move on from the circuit-based stuff, and now we're going to get into more of behavior. So this is going from the brain and looking at a whole mouse and looking at its behavior. So here we have this, what's called NF123A mouse. It's another different mouse model. The 23A part isn't really important, but they've been used by several labs in the past to study specifically NF1 associated cognitive impairments. And we're using them in our lab for the same to the same capacity. So what we're doing here is we're going to use a task which is called conditioned fear. And so in these mice, it tests this sort of domain of cognition called spatial memory, which is something that's been reported in NF1 patients to be impaired. So we're sort of testing this area of cognition, and what we do is we take the mice and put them into an environment, and we give them this stimulus that they don't really like. It's some foot shocks, and they're not really happy about it. So what happens is after we give them those little foot shocks, we take them out of the box. The next day we'll put them back in that same area. And what they should do is they would associate that area with receiving those shocks, with that unpleasant experience. And then they will respond by what is their sort of fear response by freezing. And we can measure that freezing response as a way of determining how well do they remember the space that they're in from yesterday when we originally gave them that stimulus. So we've seen that these NF1 mice show impaired spatial memory, and you progress one more so that graph comes up. Yeah, there we go. So we can see here on the graph on the right, that green bar is what we're interested in. So that's that NF1 23A mouse, that they freeze or they remember their context significantly less than mice that do not have NF1, have that NF1 loss. And what's really interesting is if we give these mice that same drug I was talking about before, the VU288 drug, that they actually will freeze very similar to the level that the non-NF1 mice do. So giving this drug is really correcting that sort of spatial learning and spatial memory impairment that we saw in the NF1 mice. And now we're really trying to expand on this and in all of these areas, really, and digging deeper into all of these mechanisms and using different approaches, really because we think that further understanding this connection between mGlu7 and NF1 allows us to sort of optimize our approach and really lead the way in what could potentially be a new approach to treat these cognitive symptoms. And with that, that's the end of our presentation, and we can take questions.
It looks like there's a couple of questions in the chat, and then we can give folks a chance to raise their hands. One was a question earlier in the presentation asking, so is there a way to tell if the learning disability is active in adults? That was the first question.
I think that is true. However, I think that a clinician like Dr. Moots is much better suited to tell us about that in the NF1 population.
The other is a genetic question, and that is, how do you give mice NF1?
Right. So there are lots of things that we can do in mice where we can mimic various human diseases. In this case, the line that Harrison talked about, where he was looking at the electrophysiology, so that in the hippocampus. So that line, one of the copies of NF1 has just been deleted from the mouse. There are other models that have specific point mutations in the NF1 gene that might mimic, some patient mutations that develop. The 23A mouse that he showed, where we showed the behavior, that mouse is missing just one part of the coding sequence of NF1. So there are lots of different NF1 mouse models out there, and some of them are much better at modeling the neurofibromas or the optic gliomas, and others are better for modeling the cognitive deficits. So we try to use a range of them so that we can, we're not just looking at one aspect of NF1, because we realize that the clinical population is very variable.
Great. We also have two questions that are very similar to one another because they're asking about pipeline. And so one of them is just sort of asking about what does the process look like, taking research and translating it into more clinical oriented use. And the other is really asking about timelines. When for this particular project, do you see that getting past phase 2 clinical trials and in something closer to human use?
Right. So, when we first started the Warren Center, we were much more dependent upon a pharmaceutical company to take the project and then go to the next steps, which are, there's a lot of things that have to happen before you can test a drug in people. right? So I work a lot with chemists here in the Warren Center. And so a chemist at their lab in our Cool Springs site who makes me a very, very small amount of compound is not going to be what we need to go into a clinical trial, right? So we're going to have to make a ton of compound. We're going to have to make sure that the compound is safe. And so as we have done this, we've gotten shorter and shorter with our timelines in terms of the time that we need to go into a clinical trial. So for one of our projects, it was about 3 years between when we started working with a company and then we moved into a clinical trial. The basic science for this target has been going on for quite a long time, and we do have a drug discovery and development program around mGlu7 now. It's always a question of funding, and a lot of our funding comes from the National Institutes of Health, and a lot of that's what we need to drive the basic science and then really figure out what type of compound that we need. And originally, earlier in this process, I thought I knew what type of compound we needed, and then we made some compounds that look like that. And now we've realized that we need to change some things about it in order to have the best chance at efficacy. So there's no hard and fast rule for how long this takes. But we have done it. We've gone, as I say, we've gone from, basic initial development of compounds into phase 1 clinical testing within 2.5 years to 3 years.
Great, thank you. If there are any more questions, feel free to either put them in the chat or you can raise your hand and we'll give you a chance to ask that question directly.
Okay, thanks everybody.
It does look like we have a few in the Q&A. One of them is, if my son had this genetic test, can we use that to see if he has these minor alleles that may be helped by this research?
Yeah, so that's really interesting. So we don't know why having the minor allele of these SNPs might have anything to do with cognition yet. So we're hoping that if we can mine data from BioView or from all of us, that we can start to understand that. I don't know why. So based on where these SNPs are in the gene, we're not really sure what they do to the gene. Originally, we thought maybe they decreased the function or the expression of the gene. And we knew from other mutations in people that cause these severe neurodevelopmental disorders that having mutations in this gene is not good. So I don't know that we're at the stage now where there would be a clinical test to segregate people based on that. It's my hope that someday we might be able to do that, and that maybe, in addition to NF1, because of the data Harrison showed in just typically developing kids, which, always learning disabilities and ADHD, that's a spectrum, right? And so there may be indications beyond NF1 that that we could move into in the future. I see there's a question about, will clinical trials test age ranges? A lot of clinical trials, the initial clinical trial is often done in adults because it's easier to get regulatory approval to do that than it is to test something in children, unless that's the main patient population. And then there's certain things that need to happen in the drug development process in order to be able to test a compound in kids. But since there are, I think it would be very interesting to look. So here's a question. We're relatively new to the NF field in general, but is there an NF1 natural history study that relates to cognitive issues? And, can we get a sense of what might be the ideal patient population based on that?
It's a great question. And there are some natural history studies, and there's a large registry. And in fact, one of the chat comments had to do with the registry that's ongoing, and a lot of groups are contributing to that's directed by the Children's Tumor Foundation. And yet what's in there probably wouldn't tell you the answers to the detailed kind of questions you'd want to know about over time as a natural history, as somebody's 5 and 10 and 15 years of age, how do they change functionally? That's a big, big question, and I'll kind of touch on that a little bit. But I would jump over just briefly to say adults who have had major problems with learning disabilities as children often still have some of those traits, that they do persist into adult life. There are some people with milder versions with NF that mature enough that those are no longer functional or significant issues for them in everyday life. And yet, I think if you could test carefully enough, you might find those were kind of persistent. Does that answer the question you had, Colleen?
Yeah, no, I think so.
About the natural history stuff?
Yeah, it's a great question. So, for Rett syndrome, we do a lot of work with Rett syndrome, and there's a very extensive natural history study where they've been following people for many years. I mean, but it gets into, I'm always thinking about like, what should our clinical trial look like? Like, what types of measurements should we have? And how do we know that we engage the target? And I see some questions in the chat that are coming up about, should we ask our geneticists about these SNPs? I don't know that we're at the stage of that yet. We haven't published these data yet. We're getting ready to. And I think that we're a ways away from using this as some type of genetic criteria. But my hope is that eventually we'll be there.
And I've seen a lot of questions in the chat too about the NF registry, which is a great resource, especially for genetic stuff like this. So one of the things that kind of limited our ability with these SNPs was that our patient population we were looking at didn't really have a lot of NF1 kids. We really wanted to look directly at these SNPs in NF1, but unfortunately our population just wasn't large enough. But with something like the NF registry that has a lot of patient samples and things like that in it, that's a great resource for doing something like this. Unfortunately, that's a lot of sort of heavy computer computational work that is just not our area of expertise. But there certainly are groups out there that are working on things like that.
Right. So we got a grant from the Children's Tumor Fund, and we were successful in getting a grant from the NIH and also from the Department of Defense around NF1, which is great. We're going to the Children's Tumor Fund conference in 2 months, and Harrison was just selected for a platform presentation, which is great. So my hope is through meeting other scientists who are working in the NF1 space that we can fill in some of the gaps that we have in my lab. I'm not a computational type geneticist, and so maybe if we can pair with some of those people, we can really, expand the impact that we can have.
I would say about the registry, it's not the same as a natural history study. And I think answering some of the real detailed questions about developmental changes in thinking and learning and behaviors is going to require more natural history studies. I think also that registry doesn't include that particular, as far as I know, like blood or patient samples. It's really an online survey. So it might not be like the equivalent of BioView or anything like that. So anyway, but it's a great thing. It still has collected just a ton of data.
So I think we should probably move on to Dr. Moots. There are some questions that you've sort of touched upon as you've answered these, and I'll do my best to try to answer them privately or make sure that we send them off to the right people to get them answered. But Dr. Niswender, and thank you so much for being here. And we truly appreciate you and, excuse me, you and Harrison. I apologize, Harrison. I was trying to remember your name. We met just momentarily beforehand. But it's a pleasure having both of you here. Thank you so much for your time.
Yes, thanks. I'm happy to be here.
Yeah, thank you.
Of course.
Look forward to the rest.
Well, speaking of the rest, we're going to move on to Dr. Paul Moots. We are blessed to work closely with Dr. Moots at NF Network, as he works very closely with NF Network down in Nashville. Dr. Moots is president of the board of the NF Tennessee, as I just mentioned. He's also a board member of NF Network. He's also a neurologist and neuro-oncologist and a former director of the NF Clinic at Vanderbilt University Medical Center. He's here today to discuss some trends and strategies to reduce the barrier to clinical trial participation, which is a huge piece of what we do and how we have seen advancement in not just research, but more importantly, clinically oriented research and potential treatments. Dr. Moots, thank you so much for being here, and it's all yours. I'll bring your presentation up right now.
Yeah, thank you. Yeah, I'll say that presentation, which has a lot of kind of granular, get down to how you look at animals at different levels, from the most basic chemical to cellular to whole animal behaviors, is really kind of part of what I'm going to cover and talk about, in the sense of kind of an overview of what's happening in cognitive and developmental research, neurodevelopmental research in NF1. And it's a really exciting area. And I just appreciate you so much coming and talking about that, Colleen and Harrison. This discussion today is really a continuation of a series of webinars that started last year, and this is the first of 2006. It's really the fourth in this series, built to kind of highlight the research projects that are being kind of supported by the NF network, and the fact that there's a research program there that is the Project Dream issue that Jeff had talked about earlier. But it's given us a chance to kind of take research and how it's done in NF and try and show the community of people with NF and advocates and everything, the importance and the value and the directions that it's moving in. And it's moving very rapidly, remarkably. So anyway, here is my discussion, research trends in NF with a focus on developmental, cognitive, and behavioral research and in NF1. Jeff, if you could move to the next slide. The first part of my discussion is going to be a bit of an overview of trends based on publications in the literature and clinical trials and funding sources and how the funding has gone. And then we'll talk more about progressive developments in terms of research waves. I call it waves. You'll see why, I think. But then really to focus on these neurodevelopmental and cognitive things that you've heard so much about already. Okay. So this is from the database PubMed, which is available online. It's open to everybody. And you can go in here and type in a keyword or a diagnosis or a condition, and it will show you publications for that disorder or condition, whatever. This in the top row is from 1945 until a year ago, articles published yearly about NF1, and the middle is NF2, and the bottom is schwannomatosis. And I say NF2 because this goes way back before the current nomenclature, terminology was developed. But anyway, you can see that there's been in all of these areas a substantial increase. If you looked in any field of medicine, there would be a substantial increase. There's so many more publications now. And yet it's really remarkable, the development that's been going on. The trends are up as far as interest and research and publications in it. Next slide, please. If you look more specifically, and I did this by putting in NF1 and added a keyword, and you can see on the left the list of different keywords that I tried. This gives 4 different samplings, 2010, 15, 20, and 25, the number of articles with NF1 plus this keyword or term. And you can see oncology, genetics, and gene. These are like the heavy hitters. They, for this 15-year epic, have been the leaders in areas of research for NF1. When you look lower down, you start to see symptoms and issues of quality of life and learning and things. And these areas have taken less of the bulk in terms of research developments. And I think you'll see why as we go through. Next slide, please. Here is another way of kind of looking and what's active and what's developing in terms of research. And this is from the clinicaltrials.gov data bank of ongoing or not even current ongoing, but all trials that are registered with this, which is over 500,000 now. It's a huge thing. It's available online. And if you're interested in trials, this is a very good source to know about. But for NF1, you can see over the last 15 years, plexiform neurofibromas have increased substantially, but a long list or a pretty substantial list of other NF1 related tumors, mainly brain and nerve tumors, but a few others have all increased substantially in terms of clinical trials that are available. And this comes right with the development of effective MEK inhibitors. It's really the success of in oncology of MEK inhibitors that has been a huge driver for new NF research and for the ways people think about NF. And I'll come back to that in a while. But next slide, please. In terms of trials, again, from clinicaltrials.gov. Now, this looks at 5-year blocks of 2010 to 14, 15 to 19, and 20 to 24 in these different categories, how many trials opened in those 5-year blocks. And you see, again, the oncology levels and, really outstrip everything else. You look down the list a bit and you see under things like developmental registries, pain, and other things, it's about the same or perhaps a little bit less. And the fact that effective and successful oncology research and clinical trials have dominated the field, it's to some extent taken away some of the kind of focus on these other areas. There's really a bit of a gap that I think as time goes by will be reduced, and I think you'll see why. And so if we could proceed on. Here, just for comparison, is similar information about NF2, where tumors and the symptoms related to tumors sort of dominate the condition. And you can see kind of a level across the board over these the number of clinical trials that opened. It's a little bit hard in here. It's an era where there are still trials that are listed as NF2 that actually include NF1 patients and things like that. And so I had to subtract some of those out. But here it's a little bit of a different sequence that you see among these epics than in the NF1 version of newly begun clinical trials. So next slide, please. Now, as far as funding for NF research, and this would include basic research and clinical trials, all of that kind of thing, you see by far the federal government is the source for funding. NF1 or neurofibromatosis is one of those conditions that are kind of earmarked in that Department of Defense funding mechanism, and it's increased. A year and a half or 2 years ago, there was quite a setback for NF because it was taken off of the list. I believe it's now returned, and this year or in the coming budget, that level is up to 25 million. So really substantial. And then there are these other organizations that contribute, and now NF Network is also to funding research. So it goes beyond this, but again, the federal government is the biggest supporter and is increasing, thankfully. And next slide, then. This is another view. This focuses just on the Department of Defense funding. And two things I really want to point out. You see as the funding got into about 2005, the lower left-hand graph, that was when the NF Clinical Trials Consortium was opened, and that was money. A lot of it went to the funding of that wonderful clinical trials consortium of now, I don't know, 15 or 20 institutions that are really the jewel in NF trials research. You'll also see on the lower right that a lot of the funding is really into basic science work, a lot like what you saw Dr. Niswender and Dr. Parent are, discussing in their talks today. And less, although substantial, it's less that goes into clinical trials and clinical research. But anyway, people view NF as a special model because of what's known about the genetics and about the oncology of it as a very important model for a lot of other illnesses. And thus, more basic science work in this area is something that's highly supported. So next slide, please. So back to this graph, and the way I look at it, and this is a little bit idiosyncratic perhaps, but the publications in NF1 dating over the years, and you see right before and right after 1990, the NF1 and NF2 genes were cloned. And thereafter, there's a pretty substantial increase in publishing on these issues. And that was the start, and what continues now is the development of genetic and genomic information about NF. And now this is NF1 in this slide, but that was what I call the first wave. And everything that's come since then is kind of, based in considerable part on that. In 2010, the first MEK inhibitor phase 1 trial was begun, and work on that drug was going on for, I think, since the mid-90s. In fact, I think the first MEK inhibitor, trametinib, I think it was discovered in 95 or something like that. But after the initiation of those trials and the success that was seen, there's another dramatic increase in publications on NF1, and a lot of it is what I would call the second wave of research development, the oncology wave. So if we could move to the next slide. So what are the big trends here? Basic science, and this is both for NF1 and F2, the neurofibromin and schwannomin, their functions, these are things that have really had a big impact on progressing and promoting research. And then the success of the research endeavors, and particularly with MEK inhibitors, has made this an area that's of interest way beyond neurology and way beyond pediatrics and things like that. And here again, you'll see my views of the genome and genetics wave, which is still going on. The oncology wave, which this got kind of mentioned a little bit in the earlier discussion, but a lot of people in medicine now know about NF, and what they know is it's a cancer predisposition syndrome. And partly because that's the most effective treatment that's been developed, and a lot of the biology is linked to that. It's no longer, when I was a student, it was a neurocutaneous syndrome, a long list of those in the back of a chapter that didn't get a lot of attention. Now it's a cancer predisposition syndrome, and it's almost kind of like the developmental issues have been a little bit in the back seat, as I've said before. So the third wave, and I'm a neurologist, I see with neurological glasses, you might say, but I think the next big wave is neurocognitive and developmental issues. And in one sense, the biology for NF1 is such a good model that other areas like autism in general, learning disabilities in general, want to know what's happening, what's the biology in NF1. and how does that apply, or does it apply to their problems? It's also the case that I think this wave will begin to address, and is beginning to address, a wider set of issues than the oncology issues, and issues that really create problems in terms of quality of life and things like that. So there's a lot of reasons to want to see this move forward. So next slide, please. Let's see, where is the field now? What's happening? And, since the disease, the illness was recognized, it was also recognized that cognitive and behavioral issues were part of it. And that's like the 1870s, So, and yet it's also recognized that those features, learning disabilities, behavioral issues and things, are 30 to 50%. In recent literature, I think, as Harrison pointed out, up to 80% of NF1 individuals have, in some measure, to some degree, some features that suggest neurodevelopmental problems. And yet, it's not a diagnostic feature of NF1. And you think like optic gliomas, that's 10 or 15%, but that's a very specific diagnostic feature of NF. Here's this very common thing that is not specific enough to qualify as a diagnostic feature. It's kind of an odd paradox. In many related things like idiopathic autism or ADHD and things like that, there are a lot of overlaps and similarities to what patients with NF1 and those diagnoses experience, and yet as people have looked closer, there are some particular things about NF1 patients that might not be common among the entire group, say of people with autism, things like that. So a profile or a specific set of features that might be a little more, the picture of NF1 cognitive problems is something that people are looking hard at and starting to find. It's also fair to say there's no specific NF1 related treatment available yet that is targeted at cognitive problems. A lot of the current treatments come from these other areas, like treatments for ADHD and such. There's a lot of good advancement, and I'll make this point multiple times about progress in neurobiology, like what you saw, earlier in the presentation tonight. And just as an aside, and I think this came up in one of the chat questions as far as adults, this is an area that has lagged far behind. The developmental changes in cognition have been much bigger. There's some mixed views about what happens. It's traditionally thought people just were kind of, NF1 patients were just kind of static, and what they were like cognitively in their late teens or 20s is how things were going to stay, basically lifelong or ideally, there's important questions about that. And yet there's very little research on that, unfortunately. So next slide. To look at clinical trials as far as neurodevelopmental issues. This is back from clinicaltrials.gov. Now, this is over a 15-year epic here, and you can see considerably fewer than in the oncology world, but still, it's not like these have been ignored at all. Learning disorders lead the field, ADHD next. In autism, in the big autism trials, they generally try not to include patients with NF1 or other syndromes. That's not always been the case, but more and more, and vice versa. In studies of autism in NF1, it's likely that future trials will not include people who don't clearly have NF1. It won't be that larger pool all put together. So next slide, please. So a year and a half or so ago, in late 2024, a large group of researchers, clinicians, funding agencies, and such got together. I think this is 25 or 30 experts, really, kind of state-of-the-art people, to think through and develop a sort of a framework and try and provide some kind of an organizational sense or guide to what should be done in neurocognitive research. Dr. Gutman, who is, if you haven't heard of him, you probably should have, and almost everybody has, from WashU, one of the world's, experts in NF1 and a very great researcher, wrote up a review of this task force meeting. And this is available online. You can go, look it up. You can Google it. And that's how I found it. And so if you want to see the details, look at the article on the Cabin Task Force. But they remarkably show there's a lot of genetically engineered animal models, meaning you take the NF1 gene that's mutated and put it in an animal and have it alter the functions, either at the cell level, or either you're looking at individual cells or tissues or full animal level. Really exciting stuff, and remarkable that it can even be done. And yet, the trials that have been done that made it to humans based on findings in those models have generally not been positive, meaning they've been negative. What had been successful in animals in these models was not successful in patients. A little caveat, there are some very good treatments like behavioral therapy and cognitive training that can be used for some problems. Depression or anxiety or a host of things that are important in NF. And yet there are treatments that there isn't an animal model that can be used to guide. So a little side issue, models don't give you every answer as to what might be a good treatment. So the big questions that came up, do these models, animal models, really portray what happens biologically in NF1? It's a hard question. And then on the flip side, the way we analyze patients clinically and put them in diagnostic categories, do those categories like autism really reflect a specific or multiple specific biological changes in the brain? I mean, the question kind of goes both ways, and I think that article points it out very well. Next slide. So here's a couple, or a little bit about the specific models. There are some that are just cells in culture, nerve cells, brain cells, the support cells of the brain, glia, and sometimes they'll grow them in a petri dish, in kind of a little sphere or organoid that have some kind of connections going on. It's not a brain per se, but it's a collection of brain cells. Putting in an NF mutated gene into those, I couldn't do it, but there's a lot of people who can, thank goodness. The next level up is animal models. And again, these are all genetically engineered, and you can see the big ones, Drosophila, the fruit fly, zebrafish. These you can make hundreds and thousands and test biochemistry and drugs and things like that to alter the biology. And then you get into mammals, rodents, and there are, swine, a pig model. And the rodents, rats and mice, provide a little better in terms of behavioral testing and things like that. Remarkably, fruit flies have some behavioral testing, grooming, and some other behaviors they can do. So they're not totally unhelpful in behavioral screening, but more often it's these more mature or more evolutionarily mature animals. One thing I learned long ago and only recently had any reason to doubt it, there currently is not a naturally occurring animal model that's used in research on NF1. And it used to be they'd say there aren't any animals who get NF1. And I always had to scratch, how could it be that this mutation can happen so often in humans, but never in any non-human animals? Well, it turns out there are some Holstein cows that get neurofibromas, and sometimes familial. Even those, they haven't been able to say is the DNA actually showing an NF1 mutation. It's still the case, just to say that naturally occurring models aren't available for research. And these genetically engineered ones, we think they're great, and yet sometimes they don't give us an answer that really turns into a positive clinical trial, unfortunately. So sometimes even the best model is still only a model. Next slide. So here's a couple examples of treatments that have come from those model systems. One I'll mention, a couple on learning and memory. The neurotransmitter gamma aminobutyric acid, GABA, which sounds like gabapentin. It's kind of closely related to that. This neurotransmitter is overactive in its function in memory circuits in NF affected engineered mice. And it's easy with a relatively widely used anti-seizure medicine, lamotrigine or lamictal, to reduce the signaling of that neurotransmitter, to reduce its release. And in mice, when you do that, it makes them do better in terms of some learning models. And I think a maze was one of them. You have to think of a mouse learning a maze. How is that? What's the equivalent to that in human cognition? You know, it's a tough question. In humans, lamotrigine in NF1 patients did not improve cognitive function in older children and young, teenagers. So here was a really good result that didn't come to fruition when it got to clinical trials. You look at the MEK inhibitors and the RAS pathway that MEK is part of. Statins, the drugs that lower cholesterol, have an effect on that RAS pathway that breaks it or blocks it a little bit. And in animals, you can do that in NF1 engineered animals and have them do better in some of these learning paradigms, learning trials. And yet quite a few studies in humans with NF1 have not showed a benefit. And they're very good trials, but that didn't prove to be very helpful. It's come up about selumetinib and MEK inhibitors directly, and I think people have been reluctant to, because of the toxicities, think of this as a good treatment. There is a little bit of data in the cancer trials on selumetinib in patients that suggested maybe there could be or was some cognitive benefit. And I must say, I haven't seen that tested in animals. And I wished I'd have looked more, but I'm not aware of that. But anyway, it's a question mark. And I think Harrison mentioned from his information that wasn't an effective treatment. But I know just a little bit on that. A different problem, ADHD and attention. Here there are altered dopamine-producing cells in mice engineered with NF1, and it seems like they also have features that would be inattention and trouble with spatial recognition and communication. The drug methylphenidate, the stimulant, alters dopamine transmission, and it improves those functions in deficient NF1 deficient mice in children, whether they have NF1 or not. It often improves that. So here again, the model doesn't really tell you for sure, is that affecting because of the NF1 biology problem or not? Anyway. Okay, next slide. One of the cardinal or big ideas that came out, and this is a little bit intuitive perhaps, from the Cabin consortia or group that got together, was that these changes in biology in the brain are very early in life, and probably in utero too, but in the first few years of life, a lot of what's happening is laid out. And so if you really want to impact it at that level, before those problems have established in the brain, early detection and treatment for prevention are really ideal. And yet those are big challenges, how to accomplish that detection issue. And then the bigger, or at least as big a challenge is what mechanisms can be discovered that are treatable. So let's go on to the next slide. A couple ways have come up, and the first, recognition of early indicators of delay or dysfunction, and people have called this a neurocognitive profile, and I'll come back on the next slide to kind of fill that idea out a little bit, what a neurocognitive profile of NF1 might be. Another way that's very exciting is biomarkers for cognitive dysfunction. And if people were really good enough at understanding neurofibromin and the mutations in the NF1 gene, they'd be better at saying which ones do or do not have effects on cognition. And that's always been a murky area. The phenotypic correlations have been difficult to establish, but they're improving. There's more and more coming about. It's always been known that big deletions, maybe the whole NF1 gene is deleted. Those individuals have more severe cognitive problems. But point mutations and a smaller deletion, it's a question mark that's starting to get answered better. There are now blood markers for a number of neurological illnesses, including like the proteins that are deposited in the brain in Alzheimer's disease. And it may be that there would be markers like that released from the brain that could be assayed in some way early. Another big area is imaging of the brain, and neuroimaging is really a booming business. And just measuring volumes is one thing, but functional analysis by MRI and PET scanning to look at brain chemistry, neurotransmitter symptoms, proteins in the brain functioning. These are areas that are really blossoming and will be applied to NF for sure. I'm not sure if there are any new EEG techniques. There are new ways of analyzing EEG. What that will contribute is a little bit of a question. In the cabin thing, they think, well, if we could just monitor kids with ADHD better, like if we made them all wear a Fitbit, would we understand, how affected they are? Well, maybe. In other illnesses, wearing Fitbits hasn't always been that helpful, but those kinds of ideas, early markers that might be useful. Next slide. Now back to this neurocognitive profile. Instead of saying, here's a child with NF1 who has autism, is if there was a way to say, in terms of executive function or attention or language or other specific, more fine-grained features, if the biology of each of those could be separated out, and people would like to believe to some degree it can, there may be better ways to focus and target those symptoms and aim treatment specifically at them. So it might be if we could have an inventory or almost like a growth chart for cognitive and behavioral symptoms in early childhood, maybe early identification and maybe better matching of symptoms to underlying biology could be accomplished. Next slide. So back to I like the name, neurodevelopmental wave. I think in terms of basic sciences, understanding the correlation between mutations and cognitive problems is an area that's really ripe and developing, and I think will tell us a lot more eventually. And we've talked a little bit, it came up, I think, before too, in terms of functions that are NF-driven through the RAS and MEK pathway, as opposed to NF problems that are independent of MEK. And it's clear that there are both categories, and it's likely that the targets for them will be quite separate. So that's another area that I think we'll see a lot of development in. I've talked a little bit about GABA and dopaminergic cells and their dysfunction in NF1. Another big area is how the glial cells and the myelin formation occur and the abnormalities that occur. The connection between myelin formation and glial activity and optogliomas is something that's kind of amazing to me, but this will be a bigger area, I think. And again, the challenge of a lot of this basic science development is how does it connect to specific features that are cognitive features in humans? And then the next slide. Part two. So we talked about the basic science part. Here's the clinical science part, the things that I see and think might be the most useful. And we've talked a bit already about the neurocognitive profile. Which features of learning disabilities and ADHD and autism are really characteristic of the NF1 version of those illnesses? And back to the natural history question, it might be quite different for attention as opposed to spatial memory or visuospatial perception. The biology of those systems and the natural history of their development are probably quite different and might be important to know. In terms of studying patients, here's where the things that the models can't tell us. There's more and more ways to get at by studying NF patients directly. Neuronal networks and plasticity in the brain and things like that in particular. And I'll highlight here, and Dr. Cutting's name came up. She was part of the Vanderbilt Kennedy Center. She is part of it. She was part of the cabin consortium group that got together. And she's done some wonderful things on dyslexia and its treatment, and accompanying that, looking at functional MRI scanning of which areas of nerve cells are the most active during and after interventions with tutoring and with statins. And really, I think that's a model for working with NF1 patients that will be really important. She's also, just as an aside, one of the most recent recipients of the NF Tennessee Impact Award, and we're really excited about that. And then so the challenges, early identification, and in that cabin report, the first 1,000 days are the critical ones in their view. Developing growth charts based on cognitive and behavioral things, the neurodevelopmental inventory, so-called, and increasing specialists in behavioral and cognitive medicine. Developmental medicine's a wonderful part of pediatrics, and to get to see one is about a 6-month wait. They're almost never in the NF clinic, and yet these are the people who you wish were there day in and day out, who could say, well, here's a person who's at high risk for developing, a 2-year-old at high risk for developing a learning disability. That kind of expertise we wish could be in the clinic every day. Next slide. And I think we're heading for home here. So what are the big trends in neurodevelopmental research? I think huge things in basic and clinical research, better models, a better understanding of the models, and connecting them to specific types of NF type 1 problems. And I think more reliance on research that deals directly with patients. And early identification. It's something that we're not very good at, and yet it provides the first step into effective prevention and probably treatment also. And one other thing, and I kind of mentioned this in passing earlier, but the trials that will be done, and like those statin trials were randomized clinical trials, really well designed, but these will be trials that aren't all of autism. These will be NF1 specific trials and really trying to get every bit of information about the biology as it relates to NF1 and these kinds of cognitive problems. So they'll be really focused trials, I think. But anyway, that's my overview. Almost all of it I gleaned from Dr. Gutman's paper and that cabin report, or at least a lot of it. And I'd encourage you, if you're interested, to look at that. It's really worthwhile.
Thank you so much, Dr. Moots. It's been a pleasure to have you speaking. I've been trying to answer questions that are a little bit more general, but if there's a couple of questions that didn't get answered, we'll make sure to forward those on to you so that we can get those answers out. In the meantime, I would like to introduce Mike Sager. Mike is a patient education manager. A few questions. There are a few questions in the chat.
Okay.
I will answer that one. That is for Dr. Niswender. So this one question would probably be best for you. So as a caregiver for my 5-year-old son with NF, I feel uncomfortable during some of the conversations with doctors in front of him because he doesn't understand the meaning of my expressed concern that I have on his behalf. I want him to see me model effective conversation, but I feel he's too young for some of the complicated issues. What strategies are there for me in these situations? Would it ever be appropriate to ask my spouse to take our son out of the room to discuss alone with the doctors?
That's a very good question. And I think that's a hard one to answer. And I think it is a very subjective situation. Just based on my limited experience with my own appointments and my own children, I would say that if you're in a situation where you feel the child is not able to understand at all at this point, it's good to give them exposure, that's for sure. And it's good to have them in the room hearing conversation. And if you feel like it would be good for your wife to take them out, great. But it's also good as they grow up and they are teenagers. One of the things that teenagers need to learn to do is being able to have discussions with their doctors. I would say go back to your healthcare provider and maybe ask them about that, or you might want to discuss that with some of your other family members. But that's a very good question, and that's often a difficult situation to manage.
All right, let me just check the chat. It actually looks like there wasn't anything else in the chat, and the other questions are for some of the other doctors. So I've already shared one with Dr. Niswender. Dr. Moots, since you're here, there is a question for you. That this person says, have traditional ADHD medications been shown to be effective or ineffective in patients with NF1? You are muted, Dr. Moots, just so you know.
Sorry. Yeah. There have been some studies that show, and I think everyday practice also, that the common medicines for ADHD are effective in children with NF1 at about the same rate. It seems to me similar to what you would see in people who have ADHD that don't have a genetic syndrome or some other underlying diagnosis. So yeah, they are effective. And well, I'll just leave it at that.
All right, I think that looks like the end of the questions that we've got. There's a couple that I will answer in the text, but I just wanted to thank everybody, Dr. Niswender, Dr. Moots, Mike, as well as Harrison for joining us today. Thank you so much. This presentation, for those of you who are still with us, will be available to watch again if you like, and we will send that out to you and make it available on our website. If you would like to contribute to Project Dream, there is a link on the NF Network website where you can make that contribution. It does take a lot for us to fund this research, but we know the value of it, and any little bit that you can contribute is certainly helpful. I will also drop that link into the chat for you so that you can click directly if that's something you'd like to do. And thank you everybody for joining us tonight. Have a wonderful evening.
Thanks, Jeff. Thank you,
Jeff,
everyone.
thank you.
Good night.
Colleen, Harrison, thanks so much. Mike, wow, I enjoyed
all.
that. I rattled a little bit, but thanks a whole lot.
Good night, everyone.
Good night.